This is BBIU, Biopharma Business Intelligence Unit. We provide structured insight and strategic analysis. In a world saturated with noise, surface narratives, and politically shaped data, BBIU operates at a different level of resolution. We focus on the forces beneath events: geopolitical and geoeconomic realignment, biopharma structural dynamics, liquidity stress and financial fragility, institutional drift and epistemic erosion, the architecture of power in global systems. Our work is guided by three principles: truth, structural coherence, and verifiable inference. Every model, every report, every forecast you see here is part of the analytical framework we use to brief decision makers worldwide. If you're looking for information, there are many places to go. If you're looking for structure, welcome. Strong clinical signal, inconsistent public disclosure. Disitamab vedotin plus toripalimab in first-line urothelial carcinoma. Executive summary, revised with temporal disclosure inconsistency. The Phase 3 randomized trial of disitamab vedotin combined with toripalimab in previously untreated HER2-expressing advanced or metastatic urothelial carcinoma, published in the New England Journal of Medicine, reports large and statistically robust efficacy outcomes compared with platinum-based chemotherapy. In the intention-to-treat population, median progression-free survival reached 13.1 months in the experimental arm versus 6.5 months under chemotherapy, corresponding to a hazard ratio of approximately 0.36. Median overall survival was 31.5 months versus 16.9 months, with a hazard ratio of approximately 0.54. The objective response rate exceeded 70% in the disitamab vedotin toripalimab arm, compared with roughly 50% in the control group. The published results are attributed to a single Phase 3 study identified as NCT 05302284, which is the sole trial identifier referenced in the article. According to the publicly accessible clinicaltrials.gov record for NCT 530284, the study is listed as recruiting active and reports 2 locations, both in Beijing. The registry does not indicate study completion and does not report results. In addition, the registry lists a study completion date in 2028, which remains unchanged at the time of publication. In contrast, corporate press releases issued by the sponsor, RemeGen Co. Ltd., describe the same study as a completed multi-center Phase 3 trial conducted across 74 clinical sites in China, enrolling 484 patients and achieving mature progression-free and overall survival outcomes. No alternative urothelial carcinoma trial using the same disitamab vedotin toripalimab combination is identifiable in clinicaltrials.gov that could account for the published dataset under a different registration number. The only clearly visible Phase 3 program combining these agents outside this identifier is NCT 0598481, conducted in gastric cancer. Taken together, these facts establish documented inconsistencies across public disclosure channels, peer-reviewed publication, clinical trial registry, and corporate communications regarding execution scale, study status, and temporal alignment of the trial underlying the reported results. Specifically, the publication of apparently mature overall survival data in 2025, alongside a registry-listed completion date of 2028, is not explicitly labeled as an interim analysis in the published article. This analysis does not dispute the reported efficacy signal. It records a verifiable disclosure divergence that limits independent reconstruction of trial execution, timing, and data provenance based on publicly available records. Structural diagnosis 1. Observable surface, pre-ODP layer. At the observable surface, the system presents as a Phase 3 oncology trial published in NEJM, large hazard ratio separations in both PFS and OS, a HER2-targeted ADC combined with PD-1 blockade, a Chinese sponsor with domestically approved biologics. No publicly disclosed FDA enforcement actions. Media and market narratives converge toward the interpretation of a globally practice-changing result, largely equating publication venue and numerical magnitude with universal validity. This layer remains descriptive. 2. ODP force decomposition, internal structure. 2.1 mass M, structural density. Structural mass is concentrated. Clinical execution limited to 2 Beijing centers. Sponsor-controlled trial design and operations. Manufacturing and quality systems rooted in a single regulatory jurisdiction. This density increases internal coherence but reduces external adaptability. 2.2 Charge C. Polar alignment internally. Charge alignment is strongly positive. Sponsor incentives, domestic regulatory priorities, and national innovation narratives reinforce one another. Externally, polarity weakens. In FDA, EMA, and PMDA environments, the same alignment produces skepticism rather than attraction, as independence and heterogeneity are prioritized. 2.3 Vibration V. Resonance sensitivity. Surface volatility is low. Clean datasets, smooth curves, and narrative consistency. This reflects damping through homogeneity, not proven robustness. Sensitivity to external audit or independent replication remains untested. 2.4 Inclination I. Environmental gradient. The regulatory gradient is asymmetric. Favorable slope toward domestic approval and reimbursement. Steep uphill gradient toward Western regulatory scrutiny. The system has not yet traversed this slope. 2.5 Temporal flow. T. Time currently benefits the system. Publication and narrative consolidation precede full regulatory stress testing, allowing confidence to accumulate ahead of verification. ODP index assessment, structural revelation. The system's internal structure is increasingly legible under pressure. Dominant forces are mass and charge, with external inclination beginning to exert counterpressure. Exposure is accelerating, not stabilizing. The ODP index is therefore moderate to high, indicating partial but incomplete revelation. Composite displacement velocity, CDV. CDV is moderate and rising. Structural exposure is occurring gradually as global interpretation expands, but has not yet reached a regime shift threshold. DFP index assessment. Force projection. Internal projection potential, IPP, is evident. The drug demonstrates biological activity and clinical effect. However, cohesion and structural coherence degrade across borders due to limited independent verification and untested manufacturing portability. The system contains force, but cannot yet project it reliably into external regulatory environments. The DFP index is moderate internally and low externally. ODP-DFP interaction and phase diagnosis. The system occupies a high ODP, low DFP phase, structural exposure without commensurate projection. This corresponds to an exposed non-agent state where signal exists but remains jurisdiction-bound. Trajectory, not snapshot, defines the risk. 5 laws of epistemic integrity, audit layer. Truth, efficacy signal is real. Global trust is unproven. Reference, anchoring relies on publication authority over regulatory stress. Accuracy, mechanisms are correctly described. Execution limits are underweighted. Judgment, effect size is overweighted relative to verification depth. Inference, global conclusions exceed structural constraints. BBIU structural judgment. The system is generating a strong domestic signal that is being overextended in interpretation. The deferred adjustment is independent verification, both clinical and manufacturing. Current responses rely on authority substitution rather than structural reinforcement, and therefore cannot resolve the underlying ODP. BBIU opinion, controlled interpretive layer. Structural meaning. The case illustrates how high-magnitude results from homogeneous environments can dominate discourse before portability is established. Epistemic risk. Conflating publication and effect size with global readiness increases downstream correction costs when verification is imposed. Comparative framing. Unlike globally distributed trials underpinning other ADC-ICI combinations, this program remains geographically concentrated. Strategic implication. Non-prescriptive. Signal discovery has outpaced verification capacity. Forward structural scenarios, non-tactical. Continuation under current conditions preserves narrative stability while exposure accumulates. A forced adjustment emerges when bridge trials, independent adjudication, or manufacturing inspections are required. An external shock, clinical attenuation, or CMC deficiency would sharply increase CDV. Why this matters? Institutional lens. For regulators, it reinforces the distinction between efficacy and verifiability. For institutions, it clarifies the gap between directional validity and asset portability. For long-horizon capital, it highlights the cost of premature consensus. For strategic actors, it demonstrates how jurisdiction-bound trust constrains projection. Access and scope note. Extended diagnostics, index trajectories, and ODP-DFP phase mapping are available under BBIU institutional access. References peer-reviewed primary source. The New England Journal of Medicine, disitamab vedotin plus toripalimab in HER2-expressing advanced urothelial cancer, NEJMO 251648. ClinicalTrials.gov. NCT 05-2284, a study of RC48 combined with toripalimab. PR Newswire, Remagen, May 12, 2025. RC48-CO16 Phase 3 reached dual primary endpoints, PFS, OS. PR Newswire Remagen, Oct. 20, 2025. ESMO 2025 Presidential Symposium Communication. Remagen official website, Company News Mirror, Oct. 20, 2025. RC48-C016 post, includes enrollment center claims as stated by sponsor. ESMO Congress 2025, Oncology Pro, presentation page for DV plus toripalimab versus chemotherapy in first-line LA-MUC with HER2 expression. Independent secondary summaries, Nature Reviews Clinical Oncology, short news commentary referencing the NEJM paper. WONC Live, coverage on NMPA SNDA and trial context. Annex 1, integrated pharmacological and pharmacokinetic characterization of disitamab vedotin, toripalimab, and monomethyl auristatin E, MMAE. Technical Annex, Mechanistic, Pharmacological, and PK Reference 1. Disitamab vedotin, RC48 ADEXI. 1.1 Molecular Composition and ADC Architecture. Disitamab vedotin is a HER2-directed antibody-drug conjugate composed of 3 integrated elements: a humanized IgG1 monoclonal antibody, a protease-cleavable linker, and the cytotoxic payload monomethyl auristatin E, MMAE. The antibody backbone is engineered to maintain high-affinity binding across a broad HER2 expression spectrum, including tumors with low antigen density. The conjugate exhibits a relatively elevated drug-antibody ratio compared with early-generation HER2 ADCs, increasing intracellular payload delivery per internalization event while simultaneously increasing sensitivity to manufacturing variability. 1.2 Functional role of HER2, delivery marker rather than driver dependency. Within disitamab vedotin, HER2 functions primarily as a cell surface delivery address, not as an obligate oncogenic driver. Therapeutic activity does not depend on sustained inhibition of HER2 signaling pathways or on gene amplification. Clinical implications include activity in tumors lacking HER2 amplification, dependence on antigen-mediated internalization rather than pathway addiction, reduced relevance of HER2 signaling resistance mechanisms. HER2 defines where the payload is delivered, not why the tumor is vulnerable. 1.3 Mechanism of action. The pharmacological sequence proceeds as follows: binding of the ADC to HER2 on the tumor cell surface, receptor-mediated endocytosis of the ADC-HER2 complex, lysosomal trafficking and linker cleavage, release of free MMAE into the cytoplasm, inhibition of tubulin polymerization leading to G2M cell cycle arrest, activation of intrinsic apoptotic pathways. Following apoptosis, loss of membrane integrity allows MMAE to diffuse into adjacent tumor cells, generating a localized bystander cytotoxic effect independent of HER2 expression. 1.4 Pharmacokinetics of disitamab vedotin, ADC-level PK. The pharmacokinetics of disitamab vedotin are dominated by the monoclonal antibody component, with payload exposure determined secondarily by linker stability and intracellular processing. Administration, intravenous infusion, distribution, predominantly intravascular. Tumor accumulation mediated by HER2 binding, limited penetration into immune-privileged compartments, including the central nervous system. Plasma behavior, biphasic concentration time profile typical of IgG-based ADCs. Initial distribution phase followed by prolonged elimination. Circulating ADC remains largely intact under conditions of adequate linker stability. Clearance, proteolytic degradation via the reticuloendothelial system. Not primarily renal. Influenced by target-mediated drug disposition, particularly in settings of high tumor HER2 burden. Half-life, measured in days, shortened by high antigen sink, anti-drug antibodies, or accelerated clearance mechanisms. ADC-level PK determines exposure duration, tumor delivery efficiency, and systemic safety margin. 1.5 Safety profile. Observed toxicities are predominantly payload-driven rather than HER2-mediated and include peripheral neuropathy, myelosuppression, and gastrointestinal toxicity. 1.6 Manufacturing and CMC sensitivities. Disitamab vedotin exhibits high CMC sensitivity, particularly in consistency of drug-antibody ratio, control of unconjugated MMAE, linker stability during storage and transport, prevention of cross-contamination in cytotoxic handling suites, and cleaning validation for highly potent payload residues. CMC robustness is a primary determinant of regulatory portability. 2. Monomethyl auristatin E, MMAE. 2.1 Chemical and pharmacological class. MMAE is a synthetic anti-neoplastic agent derived from dolastatin 10, belonging to the class of microtubule polymerization inhibitors. Its intrinsic cytotoxic potency lies in the subnanomolar range, rendering it unsuitable for systemic administration outside targeted delivery systems. 2.2 Mechanism of action. MMAE binds with high affinity to beta-tubulin near the vinca alkaloid binding site, resulting in inhibition of tubulin polymerization, mitotic spindle collapse, and G2M cell cycle arrest. Activation of intrinsic apoptotic pathways. The effect is strongest in proliferating cells but may affect non-dividing cells at sufficient intracellular concentrations. 2.3 Cellular uptake and bystander effect. MMAE is low molecular weight, highly lipophilic, and readily membrane permeable. Once released intracellularly, MMAE distributes rapidly within the cytosol. Following apoptosis of the target cell, extracellular MMAE may diffuse passively into neighboring cells without receptor-mediated endocytosis, producing a local bystander effect, including in antigen-negative tumor cells. This property enhances efficacy in heterogeneous tumors but also contributes to off-target toxicity. 2.4 Pharmacokinetics of MMAE, payload-level PK. MAE does not possess a clinically acceptable standalone pharmacokinetic profile. All relevant PK considerations arise after release from the ADC. Release context: predominantly intracellular via lysosomal cleavage, secondary extracellular release following apoptosis or necrosis. Distribution: rapid intracellular diffusion, local interstitial spread, limited systemic redistribution under stable ADC conditions. Plasma exposure: low and transient when linker integrity is preserved, increased systemic exposure correlates with toxicity risk. Metabolism, primarily hepatic, mediated by CYP3A45. Elimination, predominantly biliary fecal with minimal renal clearance. Half-life, very short when free. Clinically relevant exposure depends on ADC cleavage rate, tumor burden, and cumulative dosing. Drug-drug interaction potential, increased exposure with strong CYP3A inhibitors, reduced exposure with strong CYP3A inducers. 2.5 Toxicological profile. MMA-associated toxicities include peripheral neuropathy, bone marrow suppression, gastrointestinal mucosal injury, and fatigue. These are mechanism-intrinsic and independent of antigen expression. 2.6 Resistance mechanisms. MMAE is a substrate for P-glycoprotein. Upregulation of efflux transporters may reduce intracellular exposure and contribute to acquired resistance. 3. Toripalimab. 3.1 Molecular classification. Toripalimab is a humanized IgG4 monoclonal antibody targeting programmed death 1, PD1. The IgG4 backbone minimizes Fc-mediated effector functions. 3.2 Mechanism of action. Toripalimab blocks PD1 interaction with PDL1 and PDL2, resulting in restoration of T-cell activation, increased cytokine production, and enhanced cytotoxic immune responses within the tumor microenvironment. It exerts no direct cytotoxic effect on tumor cells. 3.3 Pharmacokinetics of toripalimab administration. Intravenous infusion. Distribution: systemic, including lymphoid tissues. Limited CNS penetration. Plasma behavior: linear pharmacokinetics across therapeutic dose ranges with no clinically significant target-mediated saturation. Clearance: reticuloendothelial catabolism, not metabolized via CYP enzymes and not renally cleared. Half-life: consistent with IgG monoclonal antibodies, weeks. Drug-drug interactions: no meaningful CYP-mediated interactions. PK unaffected by concomitant ADC administration. 3.4 Safety profile toxicities reflect immune activation and include dermatologic reactions, endocrinopathies, immune-mediated colitis, and pneumonitis, with severity ranging from mild to life-threatening. 4. Integrated pharmacological and PK interaction profile. There is no direct pharmacokinetic interaction between disitamab vedotin, MMAE, and toripalimab. Each component follows a distinct disposition pathway. Functional coupling occurs at the pharmacodynamic level, where ADC-mediated tumor cell death enhances antigen release and immune priming, while PD1 blockade sustains immune-mediated control. 5. Mechanistic rationale for combination: cytotoxic sealing and immune sustainment. Disitamab vedotin demonstrates clear cytotoxic activity as a monotherapy through targeted delivery of MMAE, resulting in rapid tumor debulking and objective responses across HER2-expressing tumors, including those with low antigen density. However, this activity is intrinsically self-limiting. The limitation does not arise from insufficient potency, but from structural constraints inherent to MMAE-based cytotoxicity. Cytotoxic efficacy depends on proliferative status and intracellular payload accumulation. Dose escalation is constrained by cumulative, payload-driven toxicities. Selective pressure favors emergence of resistant or slow-cycling tumor clones. Tumor cell death alone does not generate durable immune memory. As a result, monotherapy achieves tumor control without consolidation, leading to response attenuation over time. ADC-mediated microtubule disruption and apoptosis increase antigen availability through cellular fragmentation and release of damage-associated molecular patterns, enhancing antigen presentation. This raises immune visibility of the tumor, but does not by itself ensure sustained adaptive immune engagement. Activated T cells rapidly upregulate inhibitory checkpoints, particularly PD-1, entering functional exhaustion in the absence of checkpoint blockade. Toripalimab addresses this bottleneck by preventing premature immune disengagement. PD-1 blockade sustains effector T-cell activity in the context of antigen exposure generated by ADC-induced tumor injury, extending tumor control beyond the cytotoxic window defined by MMAE exposure. This interaction represents a conditional and asymmetric synergy. Disitamab vedotin initiates tumor damage and antigen release. Toripalimab preserves and prolongs immune pressure generated in response to that damage. Importantly, this synergy does not eliminate collateral payload exposure. MMAE released into the tumor microenvironment may diffuse into surrounding cells, including activated T cells and macrophages, creating tension between enhanced antigen visibility and localized immune collateral damage. The clinical benefit depends on maintaining a net balance in which immune sustainment outweighs payload-induced immune attrition. The rationale for combination therapy is thus not insufficiency of disitamab vedotin efficacy per se, but the need to convert cytotoxic response into durable disease control without exceeding the narrow toxicity ceiling imposed by MMAE. 6. Off-target toxicity and non-target organ involvement. Although disitamab vedotin is designed to deliver its cytotoxic payload selectively to HER2-expressing tumor cells, adverse events in non-target organs are expected and mechanistically explainable. This phenomenon arises through two distinct but convergent pathways. 6.1 On-target off-tumor exposure, HER2 basal expression. HER2 is physiologically expressed at low levels in multiple normal tissues, including gastrointestinal epithelium, bronchial epithelium, renal tubules, skin, and cardiomyocytes. While antigen density and internalization rates in these tissues are substantially lower than in HER2 overexpressing tumors, they are not null. Consequently, a fraction of circulating disitamab vedotin may undergo limited internalization in normal HER2-expressing tissues, leading to localized payload release. This mechanism contributes to low to moderate grade toxicities, such as gastrointestinal symptoms, fatigue, and mild hepatic enzyme elevations, but does not account for the dominant dose-limiting toxicities observed clinically. 6.2 Off-target payload exposure, dominant toxicity driver. The principal source of clinically relevant adverse events is payload-driven off-target exposure following MMAE release. Once liberated from the ADC, either intracellularly during tumor cell apoptosis or extracellularly through secondary diffusion, MMAE loses dependence on HER2 expression and enters surrounding cells by passive diffusion, exerting cytotoxic effects based on cellular vulnerability, not antigen specificity. This mechanism preferentially affects tissues characterized by high proliferative activity or critical dependence on intact microtubule dynamics. 6.3 Organs. Most commonly affected, peripheral nervous system. Sensory neurons rely on intact microtubule-based axonal transport, so MMA-induced destabilization leads to cumulative peripheral neuropathy, the primary dose-limiting toxicity. Bone marrow. Hematopoietic progenitors are vulnerable, causing neutropenia, anemia, or thrombocytopenia. Gastrointestinal epithelium. High cellular turnover predisposes to nausea, diarrhea, and mucosal injury. Local immune compartment. Activated T cells and macrophages within the tumor microenvironment may be exposed to diffusing MMAE, leading to functional impairment or apoptosis. This represents a form of immune collateral damage distinct from immune-related adverse events associated with PD-1 blockade. These toxicities occur independently of HER2 expression and reflect the intrinsic pharmacology of MMAE. 6.4 Determinants of toxicity magnitude. The extent of off-target toxicity is modulated by local MMAE concentration gradients, payload clearance, diffusion kinetics, cellular proliferative state, and expression of efflux transporters like P-glycoprotein. Toxicity does not scale linearly with dose escalation. Instead, it increases disproportionately once payload exposure exceeds a narrow therapeutic window. 6.5 Structural implication. The occurrence of adverse events in non-target organs confirms that the therapeutic ceiling of disitamab vedotin is defined by payload biology rather than target biology. HER2 determines delivery efficiency, but MMAE determines systemic risk. This structural constraint explains why clinical development prioritizes combination strategies to extend durability of response rather than simple dose intensification. End of document. If your organization requires structural clarity, BBIU is available for private briefings and strategic analysis. To initiate contact, visit biopharmabusinessintelligenceunit.com and reach out through the institutional access channel.